In this mini lecture, we are going to be looking at principles of synaptic transmission. In particular, we're going to pay a special attention to those principles of synaptic transmission which are of importance to pharmacologists. The first point to note is that synapses can be inhibitory or excitatory. A synapse is excitatory if it increases the chance of an action potential in the postsynaptic neuron. In the case down here, you can see the blue line is the presynaptic neuron, the red line is the response in the postsynaptic neuron. The presynaptic neuron action potentials are causing an excitatory postsynaptic potential in the postsynaptic neuron. If these presynaptic inputs are strong enough or frequent enough, they will summate to finally produce an action potential. So we can say that this synapse is increasing the chance of an action potential and is therefore excitatory. Typical neurotransmitters which are usually associated with excitatory synapses, but not always, are acetylcholine and glutamate, and they typically gate sodium and calcium channels. Conversely, an inhibitory synapse decreases the chance of an action potential in the postsynaptic neuron. In this case, action at this synapse is actually stopping action potentials from arising because it's evoking hyperpolarizations. Typical inhibitory neurotransmitters are GABA and glycine, and the most common ion channel they gate are chloride ions. It has to be said that although generally ACH synapses and glutamate synapses are excitatory, there is no principle, no fundamental reason why you should not have an inhibitory cholinergic or glutamate synapse, and in some cases you actually do. Synapses are important because they're the computational unit of the brain. Through summation, two excitatory synapses can bring about the computational unit of addition. If one of those is inhibitory, we have an instance of subtraction. We can also have one synapse modulating another. And when a synapse changes its strength, we have learning. So with modulation, summation, addition, subtraction, and learning, these are the basic building blocks of the nervous system. And so you can see why synapses are an important target for many pharmacological drugs. You may be familiar with the architecture of the synapse, and the important elements are, on the presynaptic terminal, we have a number of vesicles which store neurotransmitters. These are called synaptic vesicles. We also have voltage-gated calcium channels. These are opened by depolarization and allow calcium to enter the cell. We also have presynaptic receptors and pumps which are pumping neurotransmitter back up once it's been released back into the nerve terminal and then back into the synaptic vesicles. Postsynaptically, we have a number of receptors which are associated with postsynaptic densities, and there are also reuptake pumps postsynaptically as well. Let me just remind you of what happens at a synapse. And as I go through this, think to yourself how many elements of this might potentially be targets for drugs, either to increase or decrease synaptic transmission. An action potential neurotransmitter is synthesized and stored into vesicles, and so something has to pump the neurotransmitter into the vesicle. Are there targets there, perhaps? When an action potential arrives, the depolarization causes voltage-gated calcium channels to open, and this allows the influx of calcium. The influx of calcium signals some vesicles to fuse with the membrane and release their contents, the neurotransmitter. The neurotransmitter diffuses to postsynaptic receptors, and that causes a response. But the neurotransmitter also diffuses away and activates presynaptic receptors, and that too can evoke a response. That means then that in addition to classical neurotransmission, where neurotransmitters bind to the postsynaptic target, some neurotransmitters will also bind to receptors to themselves on the presynaptic neuron, and that could potentially block further neurotransmission. So there are many sites here for potential activation or potential action of drugs. There are many receptor pathways in the brain, and you generally find neurons that synthesize and or respond to receptors forming distinct pathways in the brain. The four that you will be familiar with by the end of this course are dopamine, acetylcholine, noradrenaline, and serotonin. If we focus for a moment on the importance of these pathways by looking at just one example, which is dopamine. Dopamine projects to the striatum, which is involved in motor control. It also projects to the frontal cortex and to the limbic system, such as the amygdala and the hippocampus, and that involves in emotion. And it also projects to the pituitary and therefore affects lactation. So that means the dopamine system is the target for many different kinds of drugs. So for example, we could target the dopaminergic motor system with drugs for Parkinson's disease. Antipsychotics target dopamine projections to the limbic structures. What does all this receptor diversity and expression pattern mean for pharmacology? You've heard before that receptors can be of hugely varied composition. That means we can make specific drugs. Receptor subunit types are expressed specifically but in several sites in the brain. So although they're expressed specifically, they're not all over the place. They are in many different sites in the brain, and that means that even a perfectly specific drug will have multiple effects. Finally, receptors to different neurotransmitters actually do look very much like each other. Their binding sites resemble each other to some extent. So that means that drugs targeting one receptor, such as X, might well actually also activate other receptors.